Good morning, everyone. Thank you for inviting me to speak here today. My name is Thom. I lead the RNA therapy team at AstraZeneca, based up the road here in Cambridge. Our team touches on quite a number of therapeutic areas throughout the AstraZeneca empire. We are developing RNA platforms, should I say, including mRNA platforms to interact with all of those, or as many of those therapeutic areas, to bring drugs to market as soon as we possibly can. So I won't go into too much around the mRNA platform. Has been covered quite a lot, but just to highlight that mRNA vaccines became shown as value after the 2020 COVID pandemic in shortening the timelines to generate vaccines, to get it to market, to actually change the course of that pandemic and saving millions of lives. So the power has been demonstrated for mRNA, and particularly in mRNA vaccines. But next is using mRNA platform for therapeutic uses. And it's also been highlighted here in the last few 20, 30 minutes that mRNA as therapeutics is touching on a number of different modalities. So from going beyond vaccine antigens to therapeutic proteins, gene editing enzymes in, for example, in the rare disease space to modify genetic mutations within people's bodies. In oncology space for cancer vaccines, which is a new field that's showing promise, but also in the CAR T, and particularly in the in vivo CAR T space, what we've just touched upon, which shows huge amounts of promise at a much reduced cost compared to the ex vivo CAR T space. So I think mRNA is at a pivotal point. It's a bit like what antibodies used to be in the 90s. So we're at a pivotal point. We've shown utility in vaccines. It's going to show utility in these other therapeutic areas. I joined AstraZeneca 25 years ago in a local company called Cambridge Antibody Technologies. So the rest of my talk is going to be focused around antibodies and the use of mRNA platforms from antibodies. And I see a couple of ex-colleagues here in the audience, and hopefully I'll connect with you later. Specifically, I'm going to talk about monoclonal antibodies and antibody fragments. So traditionally, to produce recombinant antibodies, it's generally, once the antibody has been identified that's going to treat a specific disease, it takes 4 to 6 months to generate these stable cell lines to produce this antibody, and then another 10 to 12 months to grow up bioreactors and to express these antibodies, purify, and then vial up. Of course, with mRNA therapeutics, we are the bioreactors. We produce the protein within our bodies, correctly modified, post-translational modifications, so we don't have to worry about all of that aspect up front. All we need to do is to take this genetic code, make the mRNA, formulate it, and it's vialed. We can do that. We've shown we can do that with RNA vaccines in 8 weeks. That drastically cuts the timelines to getting to clinical trials for these products. Also, with the mRNA encoding antibodies, we can potentially deliver a much more complex combinations of antibodies. For example, in hospital-acquired infections, where you could use antibodies as prophylaxis treatment to stop the hospital-acquired infections, for example, MRSA, where you're having to give multiple antibodies to treat maybe multiple virulence factors on those MRSA cells to stop those cells from infecting the patients, and also to stop antigenic escape from those antibodies. That becomes very, very cost compliant, and one way around that is to generate mRNA-based mRNAs encoding antibodies that you can then give multiplexing together, significantly potentially reducing the cost of goods and making those options much more viable, commercially viable. The very simplest term of producing an mRNA therapeutic is that you have the antibody, you have the light chain and the heavy chain of the IgG. These are generally in two separate transcripts. They can be co-formulated We need to co-formulate mRNA and NMPs because mRNA, when you inject, is very labile, will be destroyed straight away. So the formulation of the NMPs will allow injection into the patient and then production of the mRNA, production of the antibody through this pathway, through endosomal escape, translation of the mRNA in the cytoplasm, secretion of the antibodies through the endoplasmic reticulum and Golgi into the circulation, which will then, it can bind to this target, in this case, MRSA, or Staph aureus cells. So, and for the mRNA, the open reading frame encodes the protein of interest. There are a number of cis-acting elements around that for delivering mRNA exogenously. All of these are required or can be enhanced. I'm not going into any details around the enhancing of these, but all of these have to be improved together to both increase the amount of protein expression from the open reading frame, but also to reduce the innate immunity from this open reading frame. So everything from the 3' cap, and this has all been shown in the literature and shown in, well, also in the cancer, in the vaccine, mRNA vaccines that have been used in the last 5 years. So optimizing UTRs, 5' 3' UTRs, optimizing the poly-A tail to all through both promote increase of expression of the open reading frame, but also potentially the duration of expression of the open reading frame. So you have to potentially dose a lot less frequently. Using modified nucleosides to reduce the innate immunity to these exogenous mRNAs. Sequence optimizing the open reading frame. So that's not only just removing uridines because they're immunogenic, or as much as possible, but also not only GC, but there are a number of AI algorithms coming on board. We have been working at AstraZeneca for the last 5 or 6 years developing sequence optimizing algorithms, which I'll talk about in the next slide or so. That really helps significantly promoting the expression of the cargo and making it more utility for different therapeutic areas. So one example is selection of the correct UTRs. So we have generated many UTR libraries, and these are generally either tissue-specific or cell-specific within that tissue. So these are all human-based UTR libraries. We then do screening of these UTR libraries in thousands of combinational formats. We identify through in vitro screening on the relevant cell types, both cell lines and in primary cells that's specific for your target cell in the host. Identify key top combinations, and then we test those in vivo. And this is where we have generally done in mice and then in non-human primates. And this is just an example of one IgG. So the prototype is, so on the left-hand side we have antibody expressed in the serum of mice after injection IV, or IM or IV on the right-hand side, 24 hours after injection. And you'll see we started off with the prototype, which is just standard UTR from commercially available, literature available UTRs prototype, and we're getting less than 1 microgram mRNA produced in the serum. And then through iterations of this optimization process, we have then gone from UTR A to UTR B, getting 50 micrograms, and then also building upon that other aspects that I've mentioned before, so novel caps, poly-A tail, sequence engineering. You can build this expression up in an additive way. And for example, here we have UTRB plus a novel cap getting 34 micrograms per mil. So that's 120 times more than we started off with. On the right-hand side, it's really significant when when you're delivering IV, because this particular UTR library was built with liver UTRs, and particularly hepatocytes, UTRs from human cells. And you can see here we're going from about 2.7 micrograms per mil from the prototype up to well over 800 micrograms per mil. And this is vastly performs anything else that's been seen in the industry. And this now means that when we're delivering mRNAs encoding antibodies, we're getting enough produced in the host to make it commercially viable. So we reduce the amount of dose we have to give, and we're achieving the therapeutic amount within the host to be able to fight those particular indications. And that will change from indication to indication, but we are now in this ballpark where we can easily achieve this. And so this makes it, for me, perhaps a game changer for some. It won't replace antibodies at all. They're recombinant produce antibodies, but there are some aspects where, for example, in this hospital quarantine settings, you can give patients a dose of mRNA encoding antibodies with half-life extension before they have their operations. They're going to be in hospital for 3 to 6 months. Those antibodies will we have shown that they stay in the circulation for the same time as recombinant antibodies, and that they will give them protection from those hospital-acquired infections for that entire period, and subsequently after being released from the hospital. So this at a significantly lower cost compared to recombinant antibodies. So this is a game changer for some antibody-based indications. So mRNA has fantastic potential as a drug modality. It's already demonstrated utility within the vaccines, so both in COVID and now also in the flu vaccine world. But it's beginning to show utility in other therapy areas. In cell therapy for in vivo CAR T, in gene editing for rare diseases. There are a number of phase 3 clinical trials now from Intellia, etc., that's showing curative, which is absolutely amazing and fantastic. And we wouldn't have said that 5 years ago. Absolutely not. So I think this is the tip of the iceberg for RNA. It can only go up, and it can only be used in more modalities and more interesting ways. Of course, already mentioned, AI. AI is having a huge impact all along the drug discovery process for developing these molecules, and it will continue to allow us to develop more novel mRNA or RNA platforms, but also get them faster to market to help those patients. Thank you.